In this session, we will discuss the T-cell receptor and other components that are involved in T-cell activation. Let's revise the structure of alpha and beta chain of T-cell receptor. T-cell receptor have variable region and constant region. As it is the constant portion of alpha chain, so it represented as C-alpha, while this is the constant portion of beta chain, so it is represented as C-beta. Now, this is the variable region or domain of alpha chain, so it is represented as V-alpha. This is the variable region or domain of beta chain, so it is represented as V-beta. This variable region helps the T-cell to detect antigen presented on the major histocompatibility complex to cell receptor. There are disulfide linkages between the chains, so they are called as interdisulfide linkage. These disulfide bonds are also present within the chain called as intradisulfide linkages, which has already been discussed in our previous session. These linkages are in variable region and as well as in constant region. This is the basic concept which has already been discussed in our previous session. Now moving forward. This is the extracellular region, means region outside the cell. This is the cell membrane of T-cell, while it's the cytoplasmic region of T-cell. Now, as structure of T-cell receptor has been understood. After these chains, there is connecting sequence. And T-cell receptor has transmembrane portion, which connects the T-cell receptor with the membrane of T-cell. Below it, there is the cytoplasmic portion of T-cell receptor. This is N-terminal, which shows amino group, while this is C-terminal, which shows carboxyl group. But these are not only components of T-cell receptor, which play a role in T-cell activation after binding of antigen with T-cell. Let's discuss them. CD3, or cluster of differentiation 3, is a protein complex and a T-cell co-receptor that is involved in activating both the cytotoxic T-cell, or CD8, plus naive T-cells, and T helper cells, or CD4 plus naive T-cells. It is composed of 4 distinct chains. Naive T-cells are those cells which has not previously exposed to the antigen. CD3 has 3 dimer. One is homodimer, which consists of zeta and zeta monomer, and remaining 2 are heterodimers. One has delta and epsilon, while other heterodimer has gamma and epsilon monomer. Let's understand their structure on cell membrane of T-cell to make it easy for us. As we have discussed, that 2 are heterodimers, which means they have 2 different monomers. Now, moving towards first heterodimer, which has gamma and epsilon, gamma monomer, having molecular weight of 25,000. It has extracellular region, transmembrane region, and cytoplasmic region. And like gamma monomer, epsilon monomer, having molecular weight of 20,000, has extracellular region, transmembrane region, and cytoplasmic region. Other heterodimer, which has delta monomer and epsilon monomer. Delta monomer, having molecular weight of 20,000, it has extracellular region, transmembrane region, and cytoplasmic region. Similarly, epsilon monomer has extracellular region, transmembrane region, and cytoplasmic region. These 2 are heterodimer. One has gamma and epsilon monomers, while other has delta and epsilon monomers. As we have studied that beside these 2 heterodimers, other one is homodimer. Now let's discuss the homodimer. It has monomers, but both monomers are same. That's the reason it is called as homodimer. It has zeta monomer, and the other one is also zeta monomer. These monomers have small extracellular region, then transmembrane region. But its cytoplasmic region is very large as compared to its extracellular region. Now, T-cell receptor has transmembrane region having positive charge, but transmembrane region of CD3 components are negatively charged. These negative and positive charges attract each other and keep these T-cell receptor and CD3 close to each other. That will also help in T-cell activation. If we look at the arrangement of these receptors from above the T-cell, this dot represents location of T-cell receptor. Two heterodimers of CD3 are present on both sides of T-cell receptor, which are gamma and epsilon, and other is delta and epsilon, while this is the arrangement of zeta homodimer. ITAM, or immunoreceptor tyrosine-based activation motif, are on heterodimers as well as on homodimers of CD3. These receptors have tyrosine amino acid, but these ITAM, or immunoreceptor tyrosine activation motif, are not present on T-cell receptor. Now let's understand how ITAM, or immunoreceptor tyrosine-based activation motif, play a role in T-cell activation. Phosphate group is added to tyrosine, or phosphorylation of tyrosine is carried out. This will be explained in this video later in detail. In this way, CD3 helps the T-cell receptor in T-cell activation. Interaction of antigen, which is MHC plus peptide with this whole complex, causes many T-cell receptor complexes to cluster together on the surface of the cell, forming an immunological synapse. There are some other components, which are CD4 co-receptor and CD8 co-receptor. CD4 co-receptor is on helper T-cell, while CD8 co-receptor is on cytotoxic T-cells, which has already been discussed. Let's understand the structures of CD4 and CD8 co-receptors. CD4 co-receptor is a single-chain molecule having molecular weight of 60,000 found on human helper T cells. It interacts with MHC class II molecules we has many times discussed. CD4 co-receptor has 4 domains which are extracellular. Domain 1, Domain 2, Domain 3, and Domain 4. Then there is transmembrane domain of CD4, and then cytoplasmic domain of CD4 co-receptor. This is C-terminal, while this is N-terminal of CD4 co-receptor. It is a single-chain molecule having molecular weight of 60,000 and found on human helper T cells. It interacts with MHC class II molecules. But CD8 co-receptors can be heterodimer, having alpha and beta monomers, or homodimers, having alpha and alpha monomers. It is a molecule having molecular weight of 75,000 and found on most cytotoxic T cells. In humans, it is composed of 2 identical chains, that is alpha and alpha, but the equivalent in the mouse has 2 different chains. These are alpha and beta. It is involved in interacting with MHC class I molecules. Because of their close association with the TCR, CD4 and CD8 are sometimes known as co-receptors. In case of heterodimers, one is alpha chain with 1 domain, then transmembrane domain, then cytoplasmic domain. Similarly, beta chain, there is 1 domain, then transmembrane domain, then cytoplasmic domain. These chains also have N-terminal and C-terminal. CD8 co-receptor can be homodimers, means these both chains can be alpha, having 1 domain, then transmembrane domain, then cytoplasmic domain. These chains also have N and C-terminal. There are intrachain disulfide bonds in CD4 co-receptor. while these are interchain disulfide bond in CD8 co-receptor, and these are intrachain disulfide bond in CD8 co-receptor. These components also help in T-cell activation. Lipid raft are functional membrane microdomain that are characterized by highly ordered and packed lipid molecules. The lipid raft contains high level of sphingolipids, including gangliosides and cholesterol. These lipid rafts are involved in T-cell activation. These are associated with T-cell receptor. Lipid raft hold kinases that cause phosphorylation of tyrosine, thus helps in T-cell activation. This was all about the T-cell receptor. In our next session, we will discuss about the phosphorylation of ITAM.